This is a monthly program sponsored by Cohen Veterans Bioscience for the brain research and systems biology communities. Cohen Veterans Bioscience is a nonprofit research alliance whose mission is to transform research in veterans' mental health through translational research. We are focused on understanding the molecular underpinnings of diseases such as PTSD and TBI to develop better treatments and diagnostics. Before I introduce today's speaker, let me walk you through some logistics. This program is being recorded and will be archived in the Cohen Veterans Bioscience YouTube channel and website. The presentation will be approximately 45 minutes in length to allow for Q&A afterwards. Please use the Q&A button to post a question. Today our speaker is Dr. Melissa Hendel. Dr. Melissa Hendel holds a PhD in neuroscience from the University of Wisconsin and completed postdoctoral training at the University of Oregon and the Oregon State University. Currently, Dr. Hendel is Associate Professor in the Library and the Department of Medical Informatics and Clinical Epidemiology at the Oregon Health and Science University, where she directs the Ontology Development Group. She is the principal investigator of the Monarch Initiative and is an active researcher in ontologies and data standards. Dr. Hendel is known for her work on biomedical resource discovery, open science, and reproducibility, and for her work on anatomy, cell, and phenotype ontologies, such as Uberon and the human phenotype ontology. It is my honor to introduce Dr. Hendel, who will be speaking on the topic of demystifying ontologies and their utility for biomedicine. Dr. Hendel.
Thank you very much, and thanks to all of you for inviting me to speak today. Today I'm going to give a little bit of, I had been asked to give a little bit of background about, what are ontologies and how can they be used in biomedicine. So there's a little bit of sort of ontologies 101. For some of you that are more technically oriented, it might be a bit basic, but hopefully it'll bring everyone up to speed on how we can build and use these ontologies for common application across biomedical disciplines. And then at the end, I'm going to end with some work that's been performed most recently by Travis Gamble, who just completed his PhD in building a PTSD ontology. Thank you. So today I'm going to talk about why we need to standardize meaning across systems, disciplines, and tools. Ontologies, what are they anyway? I'll give you an example of some ontology-based data integration for rare disease discovery, and then again, as I mentioned, some work that was done by Travis Gamble on PTSD ontologies. So, I'm sure many of you are familiar with the scenario where you simply can't find what you're looking for. This is actually a real example where a colleague of mine was searching for stool samples in the NCBI resource, NCBI biosample. And this person did a number of different queries to look for the right kinds of stool samples, searching for feces, faeces, ordure, dung, manure, excreta, stool but not feces, stool but not feces. And as you can see, the number of records that are returned that might be relevant to this person's search is widely varying depending on what term was used. So the problem here is there's no standardization on the metadata that's used to index these records, and so you really need to do all of these searches in order to be sure that you're being inclusive of all of the content that might be relevant to you. So this is too flexible. There's no enforcement of any kind of standards, and so there's no ability to really query at any sort of depth or granularity. And so this is a significant problem that we see across biomedicine, where we simply can't even find the data that we're looking for, let alone use it effectively. When it comes to defining clinical patient features, it's even harder. So many of you will recognize what these patients have been diagnosed with, but describing that to a computer is very, very challenging. And what we often do is to have tools in the EHR that involve free text or checkboxes of this sort of nature where we have abbreviations like DF, or we use single word phrases like dysmorphic or dysmorphic facies or dysmorphic features. And none of these things are really terribly standardized, and the computer sees them as simple text strings. We get a little bit better sometimes with checkboxes where we can standardize across a single healthcare system, or even in the context of sharing data across healthcare systems where things are tagged at some high level. But even still, you know, not everyone in the world would know what ASD means in all different contexts, or how severe the global developmental delay might have been for any given patient. So what is the problem? The problem is that things that are obviously similar in terms of the phenotype descriptions mean the same thing to you but not to a computer. So for our Down syndrome children, you might record that as generalized amyotrophy, or generalized muscle, atrophy, or muscular atrophy, generalized. And the computer does not know that these things all mean the same thing, even if the clinician might. So the second problem is that many publications have little information about what the actual phenotypic features are that are seen in patients with particular mutations or particular diseases. And finally, databases cannot talk to one another about phenotypes. If I want to talk about generalized amyotrophy in the context of rare disease, that might be very different than in an EHR system. So how can we help machines better understand clinical terms? So here's a term, palmoplantar hyperkeratosis, which is a clinical term, and the computer really has absolutely no idea what this means. It's a string of letters to this computer. Further, different communities use different languages to refer to the same concepts. So the problem is not one of aligning commas and doing string matching, but rather conceptual alignment. So a patient might go to their doctor referring to themselves as having thick hand skin, whereas the clinician would refer to that condition as palmoplantar hyperkeratosis. And if we go even further and look at a mouse model, that mouse model would be described as having ulcerated paws. So these are all actually the same phenotype, but have three different strings that are strings of words that are used to refer to the same phenotype across these different contexts. The problem gets even worse when we look across the diversity of resources that are out there. So here we take, on the human side on the right, we have a palm that has our hyperkeratosis term that might be described using the Human Phenotype Ontology, or HP, and associated with a particular set of diseases in the Human Phenotype Ontology Association database. And this might be related to the ulcerated paws term that has been recorded using the mammalian phenotype ontology recorded by the Mouse Genome Informatics resource. And you think, okay, well, this isn't so bad. We can align these terms across these different resources and try to use that mouse data for assisting clinical diagnostics and clinical disease discovery. But the problem is actually much more enormous. There are literally hundreds and hundreds of different vocabularies that are used to describe different phenotypes and different attributes of organisms, whether we're talking solely in human or across different model species where we might be modeling those different diseases. So now we're going to switch gears and talk a little bit about what ontologies actually are and how do we build them. So an ontology is essentially a controlled vocabulary, but a special kind of controlled vocabulary where the terms themselves are logically defined based upon their relationships to one another. And so those relationships then allow logical inference and sophisticated data queries, and I'll show you some examples of that in just a minute. And these terms are arranged in a subtype or subclass hierarchy, but can be represented in a variety of different hierarchies, such as a partonomy in the case of anatomy, for example. And they're expressed in a computable knowledge representation language, of which there are quite a few, but some of the most common ones are RDFS, OBO, or the ontology web language. And some examples that you might be familiar with are SNOMED, the foundational model of anatomy for human anatomy, the gene ontology, and fundamentally the original ontology, which was the Linnaean taxonomy of species. So ontologies are the secret sauce behind many of our favorite tools. When you go to Amazon and it's smart enough to know that if you search for X, that you might also be interested in Y, it's because there's an army of ontologists that work at Amazon and help you discover the things that you're looking for within the millions and millions and millions of records that they might have of products that might be relevant to you. And those are smart things, like knowing that the system thinks you might be interested in azure sandals when you search for blue shoes. These are things that are related to each other conceptually, and the system uses logic to help you find the information that you're looking for. Unfortunately, this has not been applied as much as we might like in biomedicine, but we are starting to get there, and there's some examples here, such as the Ingenuity pathway analysis tool that leverages sophisticated ontologies for network biology. So the complexity of vocabulary types. This is one of those cases for ontologies where you should build fit for purpose. Ontologies, and Al, I'm not sure if you can see my cursor, hopefully you can. Ontologies are at the far end of semantic precision spectrum here, where we might start with a glossary or a thesaurus. These are things that are more listy, and as you get further up this spectrum in terms of semantic precision and complexity, you also invest more time and money and have different, more complex formats for those and languages for representing those things. So what we're talking about today is pretty far up here on this spectrum. So when we know that it's going to cost a lot of time and money to build an ontology, we need to know that it's fit for purpose, that it's actually going to allow us to represent the data, classify the data, and analyze the data in ways that we know we need. And in many cases, we don't actually need that complexity in order to analyze the data. So it's a very important feature of an ontology that you've done due diligence in understanding what the requirements really are. And those requirements have to do with how connected the data needs to be and how expressive the data needs to be. So an ontology concept then means the same thing, has the same meaning in different contexts. So in this example, we have sphingolipid transporter activity, which is a gene ontology term. And we know that when we go to the mouse ecotope or the glyprot or the diabetes resource, that we're going to be able to tell that the meaning of what's being represented in those different contexts is all the same. So if I want to take data from these three different resources and use them together, I know that I can do so knowing that the things that are annotated to this particular term are going to have the same meaning in those different contexts. So here's an example of how an ontology, a very simple example of how an ontology can be used to help perform a data query. And so here, this is a portion of the cell ontology where we have a simple classification of glial cells. And you can see down here at the bottom we have two different types of astrocytes, cerebellar astrocytes and hippocampal ones, and they're both types of astrocytes, or is a astrocyte. And astrocytes are a type of macroglial cell, which is a type of glial cell, which is a type of neuron-associated cell, which is a neural cell, which is a cell. Okay, so now if we query generally any system, and this goes back to the feces example, for any of those types, such as glial cell, macroglial cell, or microglial cell, we might come back with a set of genes as being expressed in these different types of cells. So maybe different people recorded as seeing gene expression in a glial cell, different people recorded expression in the macroglial cell. And so if we query these things, if we query just on glial cell without knowing anything about this hierarchy, we're only going to get back 20 results. But if we can expand, this is called query expansion, using the relationships in the ontology, knowing that we have data attached to these lower terms that are subsumed by glial cell, now we can query glial cell with the hierarchy in place and get back 45 results. So we can really maximize our ability simply just to retrieve data based on this subsumption or hierarchical nature of data. And it's more than just the typing or the subclassing or the is a hierarchy. It's understanding the logical relationships between things beyond that. It's knowing that the hippocampal astrocyte is part of the hippocampus, which is part of the brain. So if I have data that's associated with hippocampal astrocytes and I query on the hippocampus, I also want to retrieve that based on that part relationship. And so we have a variety of different relationship types that help us better understand how to query the data. And this is just a few examples, but for example, we know that neural cells have one nucleus. We know that if you're an enteric neuron, that you're not an efferent neuron. We know that hippocampal astrocytes are capable of regulating synaptic transmission, ion homeostasis, and axon regeneration, and that they themselves have parts such as microvilli. These things all help us query the data. Here, for example, we have some other types such as regenerative cells, which are cells but are also capable of doing something. And so in doing so, we can classify data in more meaningful ways using a variety of different relationships. So here's just some very simple biological example of how that works. And this is really the fundamental basis of how ontologies are built logically. So here, if we think about the class of all things in the world, appendages, we could classify subtypes of appendages such as antenna and wings. And wings would also have subtypes, forewings and hindwings. And that's shown here both in that hierarchical view as well as in a sort of Venn diagram type of view. And you can see that appendages then are the set of all things, and it contains antennas and wings and forewings and hindwings in that sort of manner. But we can also use those relationships to support classification too. We can talk about all of the things that are part of some thoracic segment, such as leg being a subclass of all the parts of some thoracic segment. So in this case, if we really, what we're really talking about are thoracic segment parts. And if we use the logic in the ontology, we would know that a leg and a wing would classify as in that Venn diagram of all things that are part of some thoracic segment. So we can also talk about necessary and sufficient conditions for being a member of that set. So in this case, we have sense organs, and we have all things that are capable of some detection of smell. And the olfactory sense organ would classify as a sense organ that is also capable of some detection of smell. So this is a necessary and sufficient condition for being a member of that set. So here's how we're going to use that knowledge now. So here we know that the nose is a sense organ, and we have declared that the nose is a type of sense organ. And here we've also declared that the nose is capable of detection of smell. So if we now use those equivalence axioms, or the necessary and sufficient conditions that are there within the ontology, this nose would automatically classify as an olfactory sense organ. Now, this very simple example is potentially a little silly, but it really is trying to illustrate the power of the ability to classify data. Let's say I don't know what that nose is. Maybe it's a new protein. Maybe it's a part of the cell that's been fairly undescribed. Just describing its attributes will allow me to classify it in the same way as I classify other things that I might know a whole lot more about. And so it's really helpful for gap filling on our knowledge with new data coming in. Okay, so now I'm going to switch gears and talk again about how do we put this all to use, ontology-based data integration for rare disease discovery. So now that you know a little bit about the logic underpinnings of ontologies, here's how we actually use them for integrating data. And in this case, I'm going to show you some examples from our work in rare disease discovery. And this is the work of the Monarch Initiative project. So for rare disease, we're very interested in understanding what genetic variations are causing rare disease. And for rare disease, when diseases are very rare, it's sometimes very difficult to find what genes are responsible for those rare diseases. And in OMIM, for example, the Online Mendelian Inheritance of Man repository, there are over 3,000 diseases that have no known genetic basis, and there are over 7,000 Mendelian diseases in OMIM. So roughly half of the diseases that we know are Mendelian inheritance in nature, we don't know the genetic basis of. That's an enormous number. In ClinVar, the NCBI resource for genetic variation, there are 66,000 variants with no known pathogenicity. We know that they vary significantly in the population, but we don't know what they do. So the genome is sequenced, but we still really have a real challenge in interpreting what it does. So what other data can we use? So when we look at the human coding genome, that is the genes within the human genome that code for a protein or transcript. And we look to see which ones of those genes actually have phenotypic, that we know something about the phenotypic consequences of their mutation. The value of that knowledge is about 20% of the coding genes, which means about 80% of the genes in the human coding genome, we don't know the phenotypic consequences of their mutation. That is making it very challenging to diagnose rare diseases, because if you're a rare disease with a mutation in those remaining 80% of the genome, we still don't really know how to interpret that. So what's really interesting is if you look at the orthologs of those human coding genes in the top 5 most widely used model organisms, that is rat, fly, worm, mouse, and zebrafish, and you look to see of those orthologs, what do we know about the phenotypic consequences of mutation in those organisms for the orthologs of those genes. So just looking at the genes that would be in common with human, we go up to 80% coverage. That is an enormous amount of data that's available in the model organism community that can be used to help support disease diagnostics for rare disease. So now I'm going to talk a little bit about the human phenotype ontology. Human phenotype ontology is yet another clinical vocabulary, and many people ask us why in fact do we need yet another clinical vocabulary, and the answer is because this ontology is different from all others in two ways. One is that it's designed to treat the human patient the same way that we treat any other organismal subject in describing the phenotypes that we're trying to use for analysis. So we're not building this clinical vocabulary for billing, for quality of care, for cohort discovery. We're building it to simply describe the biology of the organism. And by doing so, that allows us the second feature, which is the capability to associate and define particular nodes of that hierarchy with other ontology terms, such as the gene ontology, the Uberon anatomy ontology, or the cell ontology. And so here's a little bit about how that works. So the ontology is represented as a graph, and here I've shown you a subtype hierarchy, that subclass hierarchy, where a patient or a disease would be described as a set of nodes in the graph. So here, hyposmia, abnormality of globe location, and motor neuron atrophy. And these terms are related to one another. Deeply set eyes is a subtype of abnormality of globe location, which is a subtype of abnormal eye morphology. And these terms, such as hyposmia, are all defined in terms from other ontologies, such as the gene ontology. And here, this hyposmia term is associated with 34,000 annotations from 22 other species. So we know a lot about the biology of hyposmia from other places. And that's a really powerful attribute of the human phenotype ontology that can allow us to support wide-scale use of biological data across different disciplines. We also have worked to, and this is a different approach, but essentially using a semantic approach, the ability to understand the relationships between things. We've integrated a bunch of different disease vocabularies where we need to be able to describe, rare diseases are described in very many different places. There's the OMIM resource that I mentioned earlier, but there's also Orphanet and Decipher and the medical subject headings and the disease ontology and the National Cancer Institute's thesaurus and a bunch of other smaller resources that all have slightly different content in terms of how they describe diseases. And so here we've used the actual references between them and their descriptions and their definitions to help integrate them all into one giant mashup of all of these different disease descriptions. Now that we have this resource and the human phenotype ontology, we can actually use these resources with a variety of other, thank you so much, with a variety of other sources to help us integrate data across a large swath of biomedicine. And so here these are the genotypes to phenotypes. So for genotypes, we have information about genes, variants, orthology, and organisms from a variety of different sources, both human and models, so ClinVar, comparative toxicogenomics, gene reviews, OMIM, Orphanet, GWAS catalog, Coriell, KEGG, and then you get into some of the model organism resources, but also the Human Genome Nomenclature Committee, the UCSC browser. We have associations, things like functions, interactions, expression, and pathways, and we have diseases and phenotypes. And so different information comes from different resources. These are all the different resources. Each resource is either using natively or is mapped to one of these ontologies, such as MedGen, MESH, OMIM, the vertebrate trait ontology, the cell ontology, and sequence ontology, and things for that represent genetics, anatomy, phenotypes, and diseases. And by building these bridging ontologies, such as the one I showed you for diseases, we're able to bring in data that's represented using all of these different ontologies into a large graph structure that supports the querying across all of the data that's available in all these different resources, which were uninteroperable up until this point. And so essentially, by using ontologies, we can harmonize diseases, phenotypes, anatomy, and genotypes across a large set of basic model organism research resources and human clinical resources. So what do we do with all of that? Well, here is actually the situation. We have a patient A here. And patient A has an undiagnosed disease and is being clinically evaluated for a variety of different labs and phenotyping characteristics. And the challenge is to try to figure out what is the diagnosis of this patient. This patient has microcephaly, generalized amyotrophy, optic atrophy, polymicrogyria, contractures of the joints of the lower limbs, seizures, etc. And there might be 100 different terms here used to describe this patient using the human phenotype ontology. And so what we're trying to achieve then is a phenotype profile matching using a graph matching strategy against known diseases. So in this case, this patient might have a relationship to disease D, which here has been represented using hypoplasia of the frontal lobes, which is obviously related to microcephaly but is not identical. So this is a non-exact match here. Similarly here we have generalized amyotrophy, which is related to distal lower limb amyotrophy based on the superclass muscular atrophy. And so essentially, one by one, our algorithms go and look for the best match across using the patient's phenotype profile as a representation in a graph, matching against all known diseases, and the way that they've been represented similarly, looking for the closest profile in common. Similarly, we can do that for mouse as well. And so here we have our generalized amyotrophy term, which is related to this mouse here, which was represented as having dystrophic muscle, which are both related with the super term abnormality of muscle morphology. And this goes on. And so we compare this patient profile then against all known diseases, all known model phenotypes, and then we also do, and I'm not going to show this, but we also use protein interactome data to look for diseases that might be protein interaction partners of key candidates for this patient's disease gene. The other thing that we can do by using this approach is we can not only try to use the data to match against known diseases and known models, but we can also match the people who are involved. We can match the clinician who's doing this patient's phenotyping with the model organism researcher who might have phenotyped the model mouse M. And in that way, maybe they want to study further this patient's disease and have potential models and expertise in those phenotyping assays to help that clinician with that analysis of that patient's new disease. So here's an example tool that we've built to help perform whole exome analysis. So where we have undiagnosed patient, we do whole exome sequencing and apply a variety of standard methods for genotypic analysis and filtering, so removing off-target variants and common variants, applying Mendelian filters. We get trio analysis where we also exome, and we also get whole exomes from the parents where possible. And this leads to a variant score based on allele frequency and pathogenicity, which leads us to have a set of candidate variants that are potential diagnoses for this patient's disease. But one of the problems is we still end up with quite a few, sometimes 100 to 500 different genes might be involved following this part of the pipeline. So to try to reduce the number and really focus on the ones that might be most relevant, we add that phenotype matching strategy using the human phenotype ontology and some quality assurance mechanisms that make sure that that phenotype profile using those HPO terms from the human phenotype ontology are of sufficient breadth and depth. We compare against mouse and zebrafish and other organisms. We add in this interactome data to look for guilt by association neighbors in the network. So if one of these variants is not the candidate, or if one of the most similar diseases based on phenotype is not one of these candidates, we can walk across the network and check for the ones that might be. And and when we're all done, we hopefully end up with one variant that stands out. And this toolkit is called Exomizer. And Exomizer, this is a paper that came out last year where we used the Exomizer tool in collaboration with the Undiagnosed Disease Program at NIH to rank the variants for a set of undiagnosed patients. And in this case, the patient had a phenotypic profile that had a rare platelet set of phenotypes. And this phenotypic profile was not found in the online Mendelian Inheritance in Man repository, nor in the ClinVar repository. And so, but however, it was phenotypically similar to a mouse model that had been made. And so in this case, this mouse model was used to help diagnose this patient with having a heterozygous missense mutation in the STIM1 gene. So this was a pretty exciting success for us because we were, as far as I know, this is the first time we've used model organism data to help support a clinical diagnosis in the clinic. So one of the other things that we've been trying to achieve is the idea that patients should also be able to help phenotype themselves, and this is especially relevant for the Cohen biosciences community as well, where patient-reported symptoms are really critical. And in this case, we've built it so that that human phenotype ontology is available as a translation not only in Chinese and Japanese and French, but also in plain language and layperson, so that a patient can use the same technologies as we are technically to support finding other patients like themselves, and also to contribute their own patient-generated data to the phenotyping platform that can be used in the context of that exome evaluation. So some examples here are where a patient might describe their toes as being webbed. The medical term for that would be syndactyly. And so in doing this, essentially we can now build tools for patients that can help them participate in their own analysis of their own diseases, and also to find other cohorts and patients like themselves to help with the social aspects of managing a rare disease. It has been found that it is enormously critical to find other patients that are managing similar diseases. There's a global project here, and I was asked also to talk about communities and how do ontologies relate to communities. So I talked a little bit about our project, the Monarch Initiative, but the Monarch Initiative is part of a large suite of different efforts that have all gathered forces in a project called the Matchmaker Exchange, which allows a comparison of patient genotype and phenotypes globally to support matchmaking to find other patients. And one of the biggest issues we have in rare disease is it's very hard to make a diagnosis if you have an N of 1. If you find the next family that has the same, that's an unrelated family and has the same genotypes and the same phenotypes that are the same candidate variants, then you can much more adequately make the diagnosis, as well as, of course, to support the social aspects of patient communities. So Monarch participates in the Matchmaker Exchange to help support this, and the Matchmaker Exchange uses the human phenotype ontology to help support that phenotype matchmaking. And over here on the left, we can matchmake against known diseases and known models. This is the public data. But we can also build tools that allow clinical data sharing and clinical data management, where clinicians can go compare their patients in their own system to patients in other systems using this Matchmaker Exchange process. Okay, so now, so those are some examples from our rare disease, and then the question is, how do we build such a community and use such tools in the context of communities like that for PTSD? And so I wanted to present some work that Travis Gamble, who's a PhD student in our department, has just completed. He just defended his thesis this week, so this is really fresh, hot off the presses, and he's in attendance today and can also help answer some questions. So what he did was he essentially started with an effort to create a PTSD terminology, essentially a PTSD ontology, although it's starting off more as what I might term a terminology. And this was based on some initial pilot work that was done with the VA to make gold standard clinical notes annotations to try to identify key concepts in PTSD patient clinical records. But in this case, using that knowledge and having worked and collaborated with that group, really reviewed a variety of different knowledge sources that might represent PTSD concepts, such as SNOMED, PILOTS, the UMLS, the OBO Foundry, which I haven't spoken that much about, but which is a biomedical ontologies collection of rigorously developed ontologies. The VHA, CDW annotations, a variety of clinical guidelines and literature reviews, and then performed a variety of stakeholder interviews to find out what concepts they felt were most specific to PTSD and what sorts of things they were looking for concepts that were valid, were they missing, what kind of changes were needing to be made. And then building a semi-automated knowledge acquisition system that essentially used a set of papers that were defined as case reports for PTSD in PubMed, and then running a text mining pipeline over them using the terminology resource to look for, that had been developed thus far, to look for existing terms and to try to identify new terms, and continuing to run this as much and until the terminology was saturated in terms of trying to get those terms that represented PTSD patient symptoms, and developing this PTSD terminology system. So the system then contains symptoms, which are broken down into the DSM-5 classes, such as stressors, intrusion symptoms, avoidance, cognition, arousal, etc., and therapeutic procedures, such as psychological, case management, pharmacotherapy, etc. And when he did this, he looked at the overlap in the terms that he had captured in his PTSD terminology with existing terminology resources. And you can just see some examples over here on the left. Some existing concepts that were already in terminology systems were agoraphobia, hallucinations, pharmacotherapy, cognitive therapy, but very many new concepts such as self-blame, trust issues, impact statements, stress inoculations. And this is a little bit hard to read, but if you look closely, what you see here are all the different terminologies that are already in existence that were used for this analysis up here. I don't need to go into the details of which ones are more or less relevant, but the point is that there were quite a few new concepts here in red for symptoms and for treatments related to PTSD that were discovered in this process and added to the PTSD terminology. So what this said, the take home here is that this says that concepts that are extremely relevant for PTSD symptoms and treatments are not found in any known terminology system. This poses an enormous problem for the classification of patients using such terminology systems to help identify cohorts, to help identify diagnoses, and to help identify treatments. Travis also performed a symptom cluster analysis to look for co-occurrence of certain concepts in these case reports. So looking for using the UMLS to look for these co-occurrences, and here you can see using the word cloud some of the co-occurrences that were identified in this corpus of PubMed papers. And just some example, you can see there's, trauma is co-occurring with the word combat. And I'll show you some examples of what that looks like here. So if we take that cluster analysis and we try to look at it against the DSM PTSD diagnostic criteria, which fall into one of these different categories, we can actually take a look at how well does the PTSD terminology that Travis generated compare against the UMLS, which contains a variety of those different ontology sources. And that is shown here. So this is the UMLS. So if we look at those clusters to see how well those clusters, the concepts group between clusters, we can see that this cluster here has in part avoidance. This cluster here has arousal reactivity alterations in combination with stressors. This cluster has arousal reactivity alterations in combination with functional significance, and you can see how that goes. And there aren't very many that have 3, so these two have clusters of 3 of the diagnostic criteria. But overall, we're not seeing very good classification using the UMLS. Now if you use the PTSD terminology, you can see a much more interesting pattern here where those clusters of terms that are co-occurring, how they classify in terms of those DSM criteria is actually much more sophisticated. So here, this type of PTSD patient or case report might have a lot of stressor terms co-occurring with significant contribution of avoidance terms and functional significance terms, but that's a very different pattern than we see over here, where we have a completely different pattern with arousal reactivity alterations, co-occurring with significant contributions from intrusion and avoidance categories. And so in this way, we can develop a classifier that really subtypes PTSD patients using this PTSD terminology and based on these clustering algorithms. So here's what it looks like when you think about it in the context of a clinical note. So using the UMLS, we don't really see very much in terms of the ability to text match against those clinical notes. But using the new PTSD ontology, we can actually start to see how this looks. So here we have in light blue true positives that are marked in the note. So pharmacotherapy and cognitive therapy, those are treatments. True positives for symptoms would be agoraphobia, hallucinations. We have false positives, which are things like personality. It's lighting up here. It's not understanding the whole concept. Recurrent major depression. False negatives, things that should have been found but have not been found, and essentially by using this terminology, we can actually train this classifier to better pick up these things and to learn new terms that go into the different DSM categories. So just in summary, ontologies can support common meaning in different circumstances. They can be used to aid data integration and analysis in a variety of different contexts, but in particular, we should be applying them more in the context of biomedicine. Ontology-based integration of clinical and basic science data can improve diagnostic power for rare diseases. And I've shown you just the very beginnings of our work to try to help the rare disease community by doing exactly that. Ontology-based analysis, I think another really important take-home point is that ontology-based analysis can be combined with other types of analytics to maximize the utility. It's not the only approach, but it's one that can be enormously powerful when combined with other approaches together. So, for example, for PTSD, that might involve things like brain imaging data, which would not be necessarily an ontology-based approach, but can be combined with the type of terminology system that Travis has built to maximize utility, whether it be for diagnostics or treatment selection or prognosis. And finally, Travis's work has really deeply shown that we do in fact really need a better system for representing PTSD symptoms and treatments, that we have enormous gaps in that area, and that his initial work to build this terminology system has already been shown to help further classify based on the DSM criteria patient case reports, and it'll be very exciting to see it applied clinically and see how that pans out and leads to further advancements in PTSD terminologies. So I seem to have animation on my last slide. Something weird's going on. Oops. I do want to get my acknowledgement slide back. Sorry about that, everybody. So again, I just want to especially thank Travis for his hard work on his PhD, and congratulations, Travis, you're almost there, and his supervisor, Aaron, who's been really fantastic. A lot of my group there below, who's done a lot of work on the Monarch project and the rare disease work, as well as other collaborators on the Monarch initiative at Genomics England, Damien Smedley, Chris Mungle at Lawrence Berkeley Lab, Peter Robinson at Charity, and Tudor Grotza at Garvan. And a special thanks also to Julie McMurray in particular for some of her fantastic graphic design. So thanks very much. We can take some questions now.
Thank you, Dr. Hendel. If you have questions, please type them in the Q&A box. Okay, I have my first question for you. Thank you for this great presentation. Concerning the PTSD terminology you have built, what do you think would be the best way to integrate genetic, biologic, or neuroanatomical evidence that have been associated with this complex disease?
Well, I think that's a really good question, and hopefully my example that I showed regarding the use of genomic data with the phenotype data can give you a clue as to how we might approach that. In that case, what we've done is really try to come up with a combined rubric where we apply statistical algorithms on the genomic data, and then based on those outcomes have a metric that combines the outcome of the ontology-based phenotype matching tool. So for the case of PTSD, I can imagine that those it would be certainly enormously interesting to look at genetic variation across those different clusters that Travis had identified, and see if there's any genetic correlation with those clusters. For image, same thing with the imaging data. I think looking for patterns in the images and using some of the image analysis software, categorizing certain types of patterns in the images, and then trying to look for associations with symptomology in the PTSD records, I think would be a really interesting exercise. Hopefully that answered your question. I don't know if I can see the questions somewhere.
That's all right, I'm reading them.
Okay, sorry about that if I didn't quite get that. Feel free to email me.
Are there any more questions coming in? And if not, I just wanted to say that this will be posted on YouTube and on the website, and I did see that you had your email on the first slide, so just be prepared. You may get some questions by email afterwards.
That'd be great.
I do have another question. With regards to 6 patterns of PTSD, is it built in that these are different stages of PTSD versus different paths and/or endpoints?
So this is definitely more of a Travis question than a Melissa question. However, I will attempt to answer because I don't know if he's able to type or answer in the way things are set up, but feel free to reach out to him as well. So I would say that none of that has really been done yet. It's not. It's really just looking at symptoms as a collection and how they classify according to the DSM criteria. So I think that longitudinally, so there's two parts to that. One would be looking at patients longitudinally and seeing how those classifications change over the different clinical notes for records for that patient, for a given patient. And then I think further classifying some of the actual symptom terms in terms of their timeline of standard representation. And this is fundamentally related to some really preliminary but basic fundamental biology and how we represent disease courses. And there's some work going on to essentially represent causality and temporal unfolding in disease symptoms. And I think defining models for each disease and how those unfold is going to be really critical as a reference model to look at how patients cluster against those reference models. And right now what we do for the rare disease work is we treat the symptoms as a collection. We don't actually have, we know what the temporal unfolding is, but we're not actually using that algorithmically yet. I think for PTSD, if we could do the same exact sort of thing where we classify symptoms based on severity, based on the likelihood of expressivity in a patient that's diagnosed with PTSD, and then also in the temporal ordering, how well does a patient match the expected temporal order of a set of symptoms. And those are the sorts of features that, if Travis continued his work on building this into an ontology, would be represented, and then we'd be able to classify that information in that way, but that hasn't been done yet.
Are there any other questions? We'll give it a second to see if some more questions come in. If not, you'll get 10 minutes back in your life.
I'm so on time, ready for all your questions.
Any questions? I'm just giving it a second because there's always a lag for the questions to come in.
Yeah, I mean, I certainly would, just not having traditionally worked on PTSD, and it's really, this is really Travis's and the prior students' efforts have been sort of helping guide some of the ontology considerations as they've been doing their work. It's not really my background, but I'm really interested in understanding how this community would be interested in taking some of these approaches, and especially, as we've talked earlier, how do we integrate some of the basic research data and some of the molecular data to help support a better understanding of PTSD mechanisms, which seem very fleeting.
Well, I don't see any more questions, so I want to thank you very much for a really wonderful and informative presentation. And be prepared. I am sure you will get questions by email.
Great. Okay, well, thanks, everyone. I really appreciate the opportunity.
Thank you. Have a good one. Bye-bye.